Endosomes, lysosomes and vacuoles, their structure and function. Endosomes. Endosomes provide an environment for material to be sorted before it reaches the lysosome. The three different types of endosomes are covered individually. These include early endosomes, recycling endosomes, and late endosomes. So to start, early endosomes. Early endosomes are composed of morphologically different compartments, narrow tubular columns, and large vesicles with membrane invaginations. These differences in morphology are essential for separating their distinct functions, the main function being the sorting station of the cell. Proteins or lipids may enter the cell via receptor-mediated endocytosis. This involves the uptake of receptors bound to their ligands. They are transported in a vesicle, which becomes the early endosome. The early endosome sorts the ligands and receptors into different compartments within the endosome. The receptors are concentrated into a tubular compartment, where they are recycled back to the plasma membrane by recycling endosomes, whereas the ligands are concentrated into a sorting compartment before they are released to a late endosome, and finally to a lysosome, where the final degradation will occur. This is an example using a diagram to explain the steps. We move on to recycling endosomes. They are formed by a condensed tubular network and are located close to the microtubular organizing center. The RAB11 marker is present within these endosomes. Recycling endosomes receive internalized material from the early endosomes and direct it back to the cell surface or trans-Golgi network. The main processes recycling endosomes are involved in are epithelial cell-cell adhesion, epithelial polarity, cytokinesis, and cell fate specification. Recycling endosomes can undergo rapid or slow recycling, and further information on this topic can be found here. In the diagram that follows, the red arrows show the pathway of recycling endosomes. In-depth information of recycling pathways are also included, which can be viewed on the PREZI. Finally, late endosomes. They are typically a round or oval shape, and the compartments are derived from the vacuolar domains of early endosomes. Late endosomes can also be referred as multivesicular bodies due to their multivesicular appearance. In terms of the function, late endosomes are responsible in accepting receptors that are unsuitable for recycling. They are then sorted for lysosomal degradation. They ultimately fuse with the lysosome, resulting in degradation of their endosomal contents. Ultimately, all endosomes go through a maturation process. Endosome maturation involves an RAB7 domain being generated. The maturation process causes a hybrid of endosomal compartments accumulating in vesicles. The formation of these vesicles is vital in the development of late endosomes. Another important development is the acidification of the lumen to provide the correct environment for hydrolytic reactions to occur. This degradation happens in lysosomes. The structure of lysosomes includes a single membrane-bound organelle that functions at pH 4.8. The membrane of lysosomes contains varied integral proteins to form a protective lining against the internal enzymes. The functions of lysosomes are dynamic organelles that degrade macromolecules. In terms of phagocytosis, phagocytosis is when a specialized cell engulfs apoptotic cells, pathogens, and other foreign cells. A phagosome is formed by activation of transmembrane signaling pathways. When the phagosome matures, it forms a phagolysosome, which degrades phagotized particles. In terms of autophagy, the degradation of cytoplasmic components such as obsolete organelles. This process is important for the removal of pathogens after endocytosis and cell growth. This is demonstrated in this diagram. A conventional lysosome can fuse with the membrane, repairing a cell. When cytosolic concentration of calcium 2+ increases, lysosome exocytosis is triggered, providing a membrane. Secretory lysosomes are present in cells, with specialized lysosome compartments that store newly synthesized secretory proteins. These cells include melosomes, basophil granules, and cytotoxic T lymphocyte lytic granules. Finally, we move on to the structure of vacuoles. A vacuole is a membrane-bound organelle with little or no internal structure and is formed by a fusion of vesicles. Functions. Within plants, vacuoles take up the majority of the internal space, serving a number of functions, including keeping the cell rigid and storing materials such as sap, as you can see in this diagram. For fungi, vacuoles in fungi have a wide variety of functions, including storage of small molecules and a role in osmoregulation. For bacteria, within bacteria, vacuoles mainly act as a storage area, and gas vacuoles are also present to help control buoyancy. Finally, we move on to animal cells. Specialized animal cells have vacuoles, and their main function is assisting in endo- and exocytosis, removal of cellular waste or intracellular digestion. So there we have it, structure and function of endosomes, lysosomes, and vacuoles.